Well, here we are, everybody. This is the Week 8 challenge question review video, tutorial video. Since the thumbprint lecture kind of kicked us out of class this week, as we discussed in class, I went ahead and posted the challenge questions as a PDF for you all. And then once you've worked through them, if you choose to do so, you can watch this video and I'll go through the correct answers for those questions for you here. I'm going to do this as one long video where I go through all of the challenge questions in one shot. But of course, please pause the video before I go into the next solution. If you haven't done that question yet, use the video to the best of your advantage. But just know that I'm going to be going through all of the questions in this one video. So the first one we're going to start with here, the first challenge question is, of course, the first challenge question of the group. And that's this challenge question. This is a three-point cross problem, of course, something that we've seen in practice before in class. Essentially, we have three genes, V, CT, and S. Those alleles that are dominant have a plus sign as a superscript. Those alleles that are recessive don't have a superscript plus. These are different ways of showing alleles, but still we have dominant and recessive alleles. We have heterozygosity and the initial parent. So there's nothing really new for us here. We have a female fly that is recessive, and she is mated to a heterozygous male fly. There's no sex linkage here, so this is our typical three-point cross problem, and we're simply asked to determine the map order of the genes and the map distances between those genes. So we're going to attack this the way that we always attack these. I'm going to first identify my non-recombinant progeny, and I'm going to recognize them as my non-recombinants because they are the progeny of the highest number. And then I'm going to zero in on my double recombinants, of course. I'm going to recognize those because they are the smallest number progeny. And then I'm going to compare those two, and I'm going to look for the one allele that is different. So it looks like in the non-recombinants we have all of the dominant alleles traveling together and all of the recessive alleles traveling together. Again, I caution you, that won't always be the case. When I release last year's exam 2, you'll see that the three-point cross problem on last year's exam 2 did not have all of the dominants starting off together and all of the recessives starting together, but here that happens to be the case. And so as I compare these, it looks to me like V is what is different. So if I hone in on the V allele, I see that in the non-recombinants, dominant V is traveling with dominant CT and dominant S. And now in the double recombinants, dominant V is traveling with recessive CT and recessive S. V is new to that group. V is the only one that has changed, and so V is in the middle. And I have my map. So I can erase all of this. The next rule to solving three-point cross problems is that we never, ever do the outer genes first. We're not going after this distance until the very end. So I'm going to hone in on, I suppose, the distance between CT and V, which means I'm going to ignore S completely. And again, I'm going to identify my non-recombinants, and I'm going to compare all of the progeny to that linkage pattern. Anything that matches that linkage pattern, I'm just going to ignore. But any linkage pattern between V and CT that differs from this non-recombinant pattern, that means recombination occurred between those two genes, and I want to count that. So as I go down the list, I see that a dominant V with a recessive CT, well, that's certainly different than either of the non-recombinant linkage patterns, so I'm going to count that. Dominant V with a dominant CT, that matches, so I'll ignore it. A dominant V with a recessive CT, well, that's different, so I'm going to count it. That's 73, excuse me. Recessive V with a recessive CT, that matches what we see up here, so I'm going to ignore that. Recessive V with dominant CT, that's different, so I'll count it. And then recessive V with dominant CT again, once again it's different, so I count it. So that's the end of the list for V and CT. I'm going to add these numbers up, and I get 156. That's the number of recombination events that occurred between CT and V. That's the number of recombinants between V and CT. So I then want to divide that by the total. 1200. And I get 0.13. Now, of course, we have to multiply that by 100 to convert it into percent, so that's 13% recombination between V and CT, and that's a map distance of 13 centimorgans between those two genes. And then we're going to move on, and we're going to calculate the distance between V and S, meaning that we're going to now ignore CT. We leave those outer genes for the very last. We'll play the matching game again. We still want to identify our non-recombinants. We're going to always compare to them. Any linkage pattern that matches this non-recombinant pattern represents no recombination between V and S. Any linkage pattern between V and S that's different from this pattern represents recombination between those two genes, and so we want to count it. So our linkage pattern is recessive-recessive and dominant-dominant. The first one here on the list is dominant with recessive. That's different. We'll count it. The next one on the list is the same, dominant with recessive. That's different. We count it. We can blow right by this. That's our non-recombinant. Below it we have dominant V with dominant S. Well, that matches the non-recombinant linkage pattern, so we ignore it. And then we have recessive V with dominant S. That's different. We're going to count that. We have recessive V with recessive S. That's the same as this non-recombinant pattern. We'll ignore that. And finally, we have recessive V with dominant S. That's different. So we add up all of these independent recombination events that occurred between V and S, and we get 36. That's the number of recombination events between those two genes. We want to divide that by the total. We get 0.03. which we multiply by 100, gives us 3%, or 3 centimorgans. That's our map distance between V and S. Now, of course, we can go ahead and calculate the distance in map units in centimorgans between CT and S by just adding the two that we've already computed and get 16 centimorgans. But we have a way that we can check to make sure that that prediction is true. We can verify that we're doing this correctly. So this time we're going to ignore the middle gene because we're trying to compute the genetic distance between CT and S independently. And we're going to play the matching game. But this time and this time only, because we're doing the outer genes, we have to keep in the back of our mind that the double recombinants will match the non-recombinants. When we have a double recombination event, that means we have recombination between CT and V, and between V and S, and that switches the outer genes back to their non-recombinant linkage pattern, even though it leaves V in the middle changed. So we want to keep that in the back of our minds. The double recombinants represent two recombination events between CT and S. So we'll play our matching game. We have the non-recombinant linkage patterns of recessive with recessive and dominant with dominant. First one on the list here is recessive CT with recessive S. That matches. But it matches solely because that's our double recombinants. That means two recombination events occurred. We double those. Here we have single recombinants, 14 single recombinants. The pattern does not match. We count that once. We know that this is a non-recombinant. We'll blow past it. Here's a single recombination event between CT and S+. It doesn't match. We count it. This doesn't match. We count it. The next one doesn't match. We count it. These are all single recombination events between CT and S. And then the last one matches, but of course, just like before, that's a double recombinant. That means two recombination events occurred for every progeny there. We'll count that one twice. And we add all of these up. And we get 192. Total recombination events between CT and S in all of these progeny. We divide that by the total, and that gives us a value of 0.16, which times 100 gives us 16% recombination, or 16 centimorgans, exactly what we predict, and we know that our map is 100% correct. So another 3-point cross problem for you to practice with, but that's how you work through it. So as I said, we'll just keep going here with the challenge questions. I'll bring up the next one in just a moment. This one doesn't have any text with it, so my apologies if just looking at this diagram doesn't make a lot of sense. But what I would have said in class is that these represent chromosomal rearrangements. And what I would ask you to do is to compare this set of chromosomes with this one and name or categorize what chromosomal rearrangement occurred. Then compare this one with this one and name what occurred there, and finally this one with this one and name what occurred there. So I'm going to go right ahead now and name these, but now I've given you the instructions, feel free to pause the video and see what you come up with before I give the answers. So if we look at the first two sets of chromosomes here on the left, we'll see that it looks like something has moved. It looks like ABC has shifted to the non-homologous chromosome to its right. You see that there? But D, E, F, G, H, I remains unchanged. And even on the second chromosome, U through Z remains unchanged as well. So that seems to be the only thing that occurred, was that a piece of the first chromosome snapped off and moved on to the non-homologous second chromosome. No DNA moved in the opposite direction. So we're going to go ahead and call this a non-reciprocal translocation. For the second one, again, we're just going to take a look at these, see what we see. Well, it looks like we have movement again. Once again, it looks like ABC has moved over to the second chromosome. But this time it looks like UVW has switched places back to the first. In other words, the top three regions of the first chromosome broke off and moved to the second non-homologous chromosome, and the top three regions of the second chromosome broke off and moved to the first. We still have XYZ here below, so that didn't change. We still have D through I below here, so that didn't change. It seems to be the only thing that happened. Well, if we have DNA moving between two different non-homologous chromosomes, in other words, they're exchanging DNA information or genetic information, that is a reciprocal translocation. My handwriting is worse than usual here, my apologies. So we have a non-reciprocal translocation here in the first example, and we have a reciprocal translocation here in the second example. And finally, for the third, just take a step back, look at these for a bit. Well, here, it looks like we're only dealing with one chromosome, so no translocations at all. All of this seems to be the same between the two. In fact, it just appears that we've gained additional information, we've doubled some information, or we've duplicated it. So we're definitely looking at a duplication. But then we want to think a little bit more about that and say, well, we know that there are two types of duplications. There are displaced duplications and tandem duplications. Displaced duplications occur when the duplicated material occurs elsewhere in the chromosome, whereas a tandem duplication is when the duplicated material occurs immediately adjacent to, either right before or right after the original. And that's what we see here. The duplicated AB is immediately adjacent to or in tandem with the original. And so that's what this is. This is a tandem duplication. That's the right answer for these. Now, one of my favorite questions, in fact, of the whole semester, is the next one, because this has real clinical relevance. This is the kind of thing that a genetic counselor might do. This is a real human karyotype that you see here. Each homologous pair of chromosomes has been fluorescently labeled with its own unique color. Now, the labeling is not perfect, of course. We have some deeper and lighter regions, but for the most part, the colors are different for each homologous pair. Then those pairs are arranged in a karyotype. So these are mitotic chromosomes. They are about to divide, so they're very, very tightly wrapped, easily visible under a light microscope or a fluorescent microscope. And these chromosomes have been stained, fixed, meaning so that they don't fall apart, they don't degrade, and then arranged in size order. So the chromosomes are numbered in their size order. Chromosome number 1 is named number 1 because it's the largest pair of chromosomes that we have. Chromosome 22 is the last non-sex chromosomes because they are the itty-bittiest. And so we see that they're also arranged in numerical order, which means they're arranged in size order here. So this is a real human karyotype. We can instantly identify the gender of this organism, of this individual, I should say. This is a male because they are XY. Pretty interesting there. And there's a true genetic abnormality here. There's a chromosomal rearrangement, and I'm asking you to identify it, to diagnose this chromosomal rearrangement for the patient. So play the role of the genetic counselor, take a look at this karyotype, and determine what's wrong with the individual who has these chromosomes. So I'm going to move right ahead to the explanation. If you haven't seen it yet, feel free to pause the video until you think you know the answer. But the correct answer here is that this is a reciprocal translocation. In fact, the full diagnosis would be reciprocal translocation 922. In other words, DNA has broken off of chromosome 9 and moved to chromosome 22, and a similar region has broken off of chromosome 22 and moved to 9. If we let the colors be our guide, we see that here, on chromosome number 9, we see this out-of-place, very light violet staining pattern on the bottom tip or the telomeric region of chromosome 9. That DNA doesn't belong there. If we look for the violet chromosomes, we can see that the violet chromosomes appear to be here, chromosome number 22. So DNA moved from chromosome 22 to chromosome 9. But what else do we see? We see a very distinct and out-of-place orange chunk on the bottom of chromosome 22, and that deep orange, that's exactly the color of chromosome 9. So we also see that DNA moved from chromosome number 9 to chromosome number 22. We have an even and equal swap of genetic information between two non-homologous chromosomes. That is a reciprocal translocation, and that's the correct diagnosis for question number 2. Moving on to question number 3. Question number 3 talks about position effects, and this sets the stage for us a bit on what we will be talking about right after the exam. So as soon as exam 2 is done, we move into molecular genetics, and then we start talking about genes, how genes are turned on, the proteins they encode for. We really put a toe into the pool of molecular biology. And question 3 has some of that in it, although it's dealing with chromosomal rearrangements, the topic of this week's lectures. So let's bring those two things together and talk about position effects. So in a nutshell, position effects, if we were to loosely or informally define it, is when the function of a gene is impacted by its position, by where it is found in the chromosome. So this question is really asking, why does the function of a gene change or potentially get interrupted or worse when dealing with chromosomal rearrangements like translocations and inversions, but really the function of a gene does not change much when we have duplications? Well, first, what we have to do is consider the anatomy of a gene. And yes, genes have anatomy. So if we imagine that this straight line just represents DNA, and yes, that DNA technically is double-stranded and anti-parallel, like we talked about early on in the course, but let's keep it simple. Let's just say that this single line represents double-stranded DNA. A gene is a region of that DNA, a specific region of that DNA, that encodes the instructions for ultimately building a protein. And then that protein is going to go off and do some type of cellular job. So really that's all DNA is. It's a collection of instructions for building all of the proteins that we might ever need to make in order to survive. And then those proteins go off and get everything done. You're alive right now, you're conscious right now, you're breathing right now and thinking right now, you're writing right now and listening right now. Everything that's going on physiologically right now with you is the work of proteins. But those proteins had to come from somewhere, they had to be built, and the instructions as to how to build those proteins, they came from your DNA. That's why we call DNA the blueprints of life, because they are actually truly the instruction manual for life, but it's the proteins that are actually doing everything. So this rectangle that I've indicated here, that's a gene, or to be more precise, that's the coding region of a gene. And what's being coded for, the codes, once they're decoded, those codes are for building proteins. But genes also need an on-off switch. Some genes are needed all the time, some genes are needed hardly at all, some genes are needed in the face of a stress or a challenge, other genes are needed at certain points in development or in your lifetime. So genes need to be controlled or regulated, so they need an on-off switch, and we typically refer to that on-off switch of genes as a promoter. So now, for all intents and purposes, we have a complete gene diagrammed on the screen. We've got our promoter region to control the gene. We've got our coding region, which is the actual instructions for building the protein. When the gene is on, it's expressed, the protein is made, and the job is complete. When the gene is off, it is not expressed, the codes are never accessed, the protein is never made, and that job is not done in the cell. And that's it, kind of simply put. So let's go back and consider what we mean by position effects and how that implicates itself, or how that affects this gene. So first, let's imagine a duplication. So let's imagine any duplication of any region. Let's even make it kind of random and intrusive. Let's imagine that this region here is duplicated. And let's make it a displaced duplication. Let's say that this duplicated region actually moves over here. So now we have duplicated DNA. Let me see if I can draw this. This duplicated region here, represented by the dashed box, is the same DNA that we started with here. Now, certainly this duplicated DNA is not going to do anything. It doesn't have an on-off switch. It's not the full coding region. It's not going to code for a protein. But is the original gene in any way impacted? Is this gene that we started with no longer functional because a part of it has been reproduced or duplicated over here? No, it looks to me like this gene remained completely intact. No position effect at all. It's still where it was before. The original is still intact. We haven't lost any DNA from there. We haven't moved any DNA from there. So I don't see much of a position effect whatsoever. To be sure, this DNA is never going to do anything. It's non-functional. But the DNA we started with, the gene we had in the beginning, no harm done, no problem there. So there it doesn't seem like position effect is doing that much harm. In fact, position effect is not an issue at all. But now let's imagine an inversion. And actually, let's do an inversion of pretty much the same region. So remember, an inversion means that the piece that's being inverted pops out, flips over 180 degrees or completely upside down, and then pops back in. So let's try to imagine what that would look like. Let's invert this region. So I think I've got it there. And I'll even do this just to drive home exactly how bad that is. So that's the inversion. Again, the dotted line represents what we started with. Now, if we take a look at that, I don't think that's a functional gene, do you? It looks like we broke off a big chunk of that gene, including some of the non-coding region, including some of this part that really doesn't call for the instructions for building proteins, and we've glued it all back together in a way that it doesn't go. You can think of this as puzzle pieces, jigsaw puzzle pieces that have been jammed into place in a way that they really don't fit. So this is not going to code for a protein at all. The protein coding region, the all-important region that is the actual instructions for building the protein, no, not going to cut it anymore, not going to encode that protein. So this is a very, very dramatic chromosomal rearrangement. The position of the gene has definitely been affected. We do have a position effect. This gene will be non-functional. We've disrupted it completely. So now let me restore this back to what we started with, and we'll take a look at a translocation. And we'll do a non-reciprocal translocation just to make it a little bit more simple and straightforward. And if we're going to do a translocation, let's make a second chromosome here somewhere with its own genes and its own DNA. Let's zero in on that same region just for the sake of consistency and say now it's going to be involved in a non-reciprocal translocation. That means this DNA is going to break out of its initial chromosome and insert itself here into this new chromosome. Well, first let's make some room for it because it is going to literally insert itself. And then let's do the insertion. And of course the original, this isn't a duplication, that means the original is actually gone. So this region is missing, and we have to now stitch these ends together. So I'm going to move that DNA there. And yeah, that's our reciprocal translocation. What do you think, guys? You think this is going to code for a functional protein now? Can you make a working machine when you've lost half of the instruction manual as to how to build it? No, that's not going to work. This gene will no longer be functional here. This segment of a gene certainly won't be functional at all. It doesn't even have a promoter region to drive it. So once again, we have a position effect problem. We have the position or physical location of the gene disrupted by the chromosomal rearrangement. So that's really what question 3 is getting at. It's getting at trying to get you to think about what happens to the DNA when chromosomal rearrangements occur, and specifically what happens to the genes that are encoded on that DNA. With duplications, the original is untouched, and so we don't have position effects. As long as the original is untouched, we shouldn't have a position effect at all. But things like translocations and inversions, they disrupt the original. The original is what's being manipulated. And if the DNA that's being rearranged happens to contain a gene on it, or even worse, as I've diagrammed here, if the region that's being rearranged happens to have a fragment of a gene on it, we will see position effects. Now to the last 2 challenge questions from the day. There's the last 2. So the first question is kind of a thought experiment question here, number 4. Explain why aneuploidy of chromosomes with larger number designations can sometimes result in people who live with the disorder, such as trisomy 18 or trisomy 21. Trisomy 21 is, of course, Down syndrome. Trisomy 18 we talk about quite a bit in the chromosomal rearrangement lecture. But we don't really see people that are trisomy 1 or trisomy 2. Those disorders don't exist. It's not because those trisomies don't occur. It's because individuals that have trisomies are unviable and they spontaneously abort long before birth. So why is that? Why do trisomies of the higher number chromosomes often remain viable while trisomies of the lower number chromosomes, they don't? So I'm going to go right into the answer here, but you can pause the video if you want to think about that longer. The answer here comes back to what I talked about a moment ago in the karyotype question. Chromosomes are numbered 1 through 22, and then of course the sex chromosomes, by size order. So chromosome number 1 is the largest, and chromosome 22 is the smallest. If you think about it, chromosomes that are very large have more genes on them, of course, because they're bigger. And chromosomes that are quite small have fewer genes on them. And this all comes down to that notion that we talked about in X inactivation, and we brought it up again here with aneuploidy and polyploidy, this idea of gene dosage. We are evolved to have 2 alleles in our genome for every gene. If we have less than that, we have metabolic and physiological problems. If we have more than that, we have metabolic or physiological problems. If our dose of each gene is too high or too low, there are issues with the individual. This is why X inactivation was so key. You and I, males and females, we had to have equal doses of our genes on the X chromosome. We couldn't have a species running around where half the species had 2 X chromosomes with 2 alleles for every gene, and other members of the species just had 1, so X inactivation evolved to compensate for that. Here, with aneuploidy, we might be able to skate by and have viability if a small number of genes have their dosage affected. But the more genes with disrupted dosages, the more genes where there are too many or too few alleles in a person's genome, the less and less chance we're going to have viability in that case. We see trisomy 21 and even trisomy 18 because those are the smaller chromosomes. Those are the chromosomes with the fewest genes on them, and so those are the chromosomes that you might be able to see some viability. Now granted, there are some disorders, there are some issues with those individuals, but for the most part, we see viability because the number of genes whose dosage is impacted is relatively small. Once we get to the lower number chromosomes, which are bigger, we're affecting the dosage of that many more genes. Too many genes, in fact, to see viability. Because the larger chromosomes with the smaller number designations have more genes on them, more and more gene dosages are impacted by trisomy or monosomy, and for that reason we don't see viability in those individuals. So that's really all it comes down to. The crux of this question is knowing that chromosomes are numbered based on their size, and the smallest numbered chromosomes are the largest, with the largest number of genes on them, where the largest number of gene dosages are affected in trisomy or monosomy, and that's why those individuals are non-viable. Question number 5 highlights one of my favorite stories of chromosomal rearrangements, a seeming paradox. Humans and chimps, our nearest living primate relative, were 98% genetically identical. That means we have 98% of our DNA in common, not only similar, but identical. Only 2% of our genomes differs between us and chimpanzees. Yet chimps have 48 total chromosomes and we have 46. How in the world can that be possible? How can we only have 2% of our DNA different when they have 2 more chromosomes than us? Now, even if we just go on percentages alone, 2 more chromosomes out of 50% is far more than 2%. It's closer to 5%. Now, why would we have such a small genetic difference when we have such a radical difference in chromosome number? I'm going to go right into the answer here, but certainly pause it if you'd like to think about it. The answer comes down to a very unique type of chromosomal rearrangement, a very unique translocation, in fact, called a Philadelphia translocation. Now, I don't know the history of that term, so I'm sorry I can't give us any insight as to where that name came to be, but this is a Philadelphia translocation. Essentially, a Philadelphia translocation occurs when the regions that recombine, or the regions that have the translocation, occur at the ends of the chromosomes. So if we were to zoom in here a little bit and take a look at these chromosomes, imagine that this piece of this chromosome and this piece of this chromosome swap and join together, and then the larger arms swap and join together as well. In other words, what we're saying is imagine that we have a break here and a break here, and these 2 large fragments of chromosomes are glued to one another. Well, if we do that, we would actually see that the chromosome we would get would be this one, a much longer chromosome that really represents 2 smaller chromosomes that were glued together in the translocation. That's a Philadelphia translocation. It's when 2 relatively small chromosomes translocate in such a way that the majority of the DNA from each of those 2 chromosomes links together to make a much larger single chromosome. So that happens occasionally, and it happened in the evolution between chimps and humans. You may have noticed already, but these letter codes correspond to this legend, and this is what we see for orangutan chromosomes, gorilla, chimpanzee. We see a lot of similarity between these chromosomes, really not a lot of difference, and then the human lineage. If we look at the banding pattern that we can generate using some of those stains that we talk about in the lecture, we see that the banding patterns line up. We've got these 2 moderate bands. We've got another doublet of bands here. It seems like all this DNA is very similar. Another doublet here with a very narrow non-stained region between them. We have this characteristic doublet of doublets here. We've got another one here, a very large single band of staining, and finally another doublet. Lots of homology, lots of match between all of these chromosomes of all of these primates. Not surprising, because again, we're all primates. We're all part of the same family tree. But if we take a step back, we see although all of this DNA is very similar, that DNA that we just identified, it is conserved between 2 separate non-homologous chromosomes in chimps, gorillas, and orangutans. Whereas in humans, somewhere in the evolution of humans, a Philadelphia translocation occurred, and those 2 relatively smaller chromosomes translocated with one another and joined, giving a much larger chromosome here in the human lineage. So, is the DNA that we see on these chromosomes in chimp, is the DNA sequence, are the bases of A's and T's and G's and C's nearly identical to what we see in humans? Most certainly. In fact, that DNA is 98% identical. But surprisingly, that DNA that is so similar between those two species is spread across 2 different chromosomes in chimpanzees. Whereas after the Philadelphia translocation, all of that DNA is now found in a single chromosome in humans. Now remember, this happened with homologous pairs, because even chimps are diploid. So if we look at the chimpanzee chromosomes where this occurred, we've got the short chromosome that's up top, and we've got the other chromosome that's on the bottom. But those 2 came from that chimp's mom. And then we've got the short chromosome and the larger one. And those 2 came from the dad. This is the mother's smaller chromosome. This is the father's smaller chromosome. This is the mother's larger chromosome. This is the father's larger chromosome. But in each of those cases, a Philadelphia translocation occurred, and these chromosomes joined with one another, yielding a much, much larger chromosome, again where they had joined in the human lineage of evolution. There's the join mark between them. And that's where the human chromosome came from. When I initially joined these together, when I first drew these on the screen, we had 4 total chromosomes. And now how many do we have on the screen after I've joined them together? We have 2. How many chromosomes did we lose? We lost 2 chromosomes because we do this Philadelphia translocation with 2 chromosomes with each member of the homologous pair. So if we go back to our question, how do chimps have 48 and humans have 2 fewer, 46? Well, that 2 fewer came from the Philadelphia translocation of the maternal chromosomes in the chimp and of the paternal homologous pair chromosomes in that chimp, losing 2 chromosomes along the pathway of human evolution. And that's how we can have almost nearly identical DNA while also having 2 fewer chromosomes. Pretty interesting stuff. It shows the power of these chromosomal rearrangements. So those are all the challenge questions from this week. I hope that I've given some insights as to the right answers. If you have any further questions, of course, you know how to get in touch with me. But otherwise, I'll see you in class next week for finally a normal week where we will meet on a Monday and a Wednesday.